Hi all, welcome back to Environmental Science. Last time out, we were discussing hunger, world's population, technology, whether or not, based on the number of people on the planet, we could be self-sufficient and raise enough food for all 8.2 billion people to survive. And that's highly unlikely based on the land that we have. We've already overshot that capability. So the next kind of topic is, well, if we can't have self-sufficiency, is technology that makes our ability to grow more food on a landscape, is that our next step? Okay. And I talked to you about a little bit that in the 1930s, 1 acre of land would support or could grow about 25 bushels of corn. And today, with the technological advances of GMOs, pesticides, fertilizer, on that 1 acre of land, we're growing about 170 bushels of corn. Okay, so about 150 bushels of corn more per acre today than we did in 1930. Okay, so that clearly is a huge, huge benefit, and if that benefit could be even advanced more, then the idea that we don't have enough food to support 8.2 billion people, because there's 800 million people going hungry, then maybe GMO is the answer. Maybe. So let's look at what GMOs are. So again, GMO stands for genetically modified organism. And so let's first look at kind of how they are created. So there's a lot of misconceptions around GMOs, and that's mainly because we lump them all into this heading of genetically modified organism. Okay, so these are the ways at which you can get a GMO. You can do traditional breeding experiments where you're taking one variety and mixing it with another, and hopefully it will produce a variety that you want. Okay, so that's just crossing things. That's been done, since humans became agriculturalists, and kind of the Middle East region is where we believe that it developed, when we moved from hunter-gatherers to be individuals, farmers in essence. So it's been happening for thousands of years, and create lots of different varieties. In fact, corn itself came from a wild plant called tezonite, which was just a cross that Native Americans in central Mexico, or what's now central Mexico, were just crossing the grain, and they just kept crossing it, and the kernels get bigger, and the cob got bigger, and to the point where it was more nutritious, it was easier to grow, lots of things. That's traditional breeding. Okay, there are other ways that we can do that. So pretty much every single crop that you can think of on the planet, traditional breeding, the cross between one organism and another organism, has happened. Okay, and you might not think, well, that doesn't sound like a genetically modified organism. Well, it is, because a lot of these crops would never come in contact with each other. We're taking crops from one region and another region that no way the pollen could ever travel that far, and we're mixing them. We're mixing, crops that are same genus, maybe different species, and seeing what can happen. So it's still a genetically modified organism, but there's a couple components with this. There's no regulation. There's no safety testing, anything required, no regulatory. You can mix organisms by swapping pollen to the pistil or the ovary of another plant as many times as you want, and nothing happens. That's traditional breeding, been done for thousands of years. Okay? The next one that was kind of invented was, we knew and know for a long time now, that if you zap things, embryos, pollen, ovaries, if you zap things with high levels of radiation, or you can do this chemically, you can mutate a plant's genetic material, okay, or an organism's genetic material. Those are called mutagens. Right? And so if you can mutate it and then see what happens, see what grows from that, and if it's something beneficial, if it's something that we want, then great. If it's not, it's tossed out, and another organism is zapped with high levels of radiation, or can do this chemically. And so there are quite a few crops that have come out of this, so different varieties of wheat and ruby red grapefruits, different species of apples. So there are different species that have come out of this. Okay, and again, this one's been around for not thousands of years, but it's been around for probably 100 years, maybe. Okay, I'm not sure, don't quote me on that, but there's quite a few organisms that are the result of mutagenesis. Okay? And again, there's no safety testing, no regulation required for this type of crop. You don't even have to label it. Okay? So you don't have to label traditional breeding, you don't have to label mutagenesis. Okay? These guys, there's no regulation, no safety, nothing. So you don't even stick a label on them. You've ate both of these many, many times. Okay? The next three have regulation. Okay? And the next three are, in many cases, more controlled. And the outcome is more predictable and more designed. Okay? The individuals doing RNA interference or transgenic or gene editing are trying to either take and silence a gene so it doesn't express itself. Maybe that's a toxin. Maybe that's something that's laying down like in a cuticle on the outside of a fruit or a vegetable, and you don't want that anymore. There's lots of ways. So RNA interference is a way to basically target a gene to turn it off or silence it. Now, you could also turn it on potentially, but normally we're interested in turning it off. So just turn that gene off, and then that's no longer produced, and so that organism no longer produces a toxin or something like that, and now we can consume it. And so lots of things, apples and a bunch of different things, tomatoes have all had this done to them. Okay. For the most part, I'd say the first three. Mutagenesis, most people don't know that mutagenesis occurred, and that's how a lot of these crops are generated. So because you don't have to label it. But I would say these first three, maybe RNA interference is less problematic from a society's opinion of a genetically modified organism. If they knew that you're just going in and turning off a gene, and that's what you're doing, that is less problematic. The one that is often got a lot of the most criticism is transgenic. Okay? And that's when you're taking genes from another organism and you're inserting it into an organism. So that's a transgenic response. So you take one gene from a different organism, or maybe a different variety. So it could be the same species, just a different variety. But normally what we're talking about is a different organism that produces a gene, and you're taking that gene and you're sticking it in to a crop. Okay? And this here is how we get things like Roundup ready corn, soybeans, where we found species of weeds, typically, that produced a gene that allowed them to resist the effects of, say, things like Roundup. Then we take that gene and we could insert it into a crop. Then you could spray your field of crop with Roundup and it doesn't die, but the weeds do. Okay? And so that is a transgenic mutation, or a transgenically modified organism. Okay? And lots of species of crops currently have that. These two are highly regulated. There's lots of testing going on, and they need to be labeled as GMOs. The last one is the newest kind of technique, and that's gene editing. It's very, very similar to transgenic, except for you don't have to insert another organism's DNA. You can just change it. You just alter, you engineer that. So you can go in and you can say, hey, there's this region, there's this gene, I know the A, T, C's, and G's, the nitrogenous bases, I know what it is that I want to fit in there. I want to remove this old gene and put this new one in. So you're editing genes. You're not required to swap genetic material from a different species or anything like that. You're just editing genes. You're cutting and pasting new genes into an organism. And this one is depending on what country, what crop, it has mixed regulations. And in fact, the USDA does not recognize, at least CRISPR, this technique of cutting and splicing and putting genes in. It does not recognize it as a genetically modified organism. So even though genetically modified organisms are highly regulated in some cases, some are not, and some are not even recognized as being genetically modified organisms by the regulatory agents that we have in the United States. Okay? And so the three that we have are the Food and Drug Administration, okay, the United States Department of Agriculture, and the Environmental Protection Agency. Okay? These three entities are supposed to regulate crops or the effects of genetically modified organisms. Sometimes they do, sometimes they don't. Sometimes they have opinions on whether something should be considered a genetically modified organism or not. Now, I think most scientists would argue, unless the name genetically modified organism means that no one's going to buy the food that you generate, but would argue that if you cut the gene out of a species and put a different gene in, even if that other gene is not known to another species, like you created it yourself, or maybe it's from another individual within a population, and you stick it in, that you've modified an organism's DNA. Therefore, gene editing is a genetically modified organism. But many examples of gene editing, according to the USDA, are not genetically modified organisms. So they don't have to label them. So if that scares you, that genetically modified organisms are being sold to you and you're consuming them, just note that for 3 of the ways at which we can do this, you don't have to even tell them. So you've ate it, you've consumed it. Whether that's good or not, it's up for debate. But what my reason for telling you this is that without genetically modified organisms, we're talking maybe 10 to 20% of the food production that we currently have. That's what we would have without genetically modified organisms. We're talking maybe 25% of the food production that we currently have. And if we already have an issue with hunger in the world, take and remove 75% of the world's food production. And because, genetically modified organisms are no longer allowed, right? And see where we're at from a hunger perspective. Okay? I'm not saying this technology is the answer for food hunger, or for hunger in the world. What I'm saying is it's pretty strong technology to increase the amount of food production we can have. And so I'll let you watch this video also. And that really just gets at, in the United States, okay, about half the people in the United States think GMOs, or genetically modified organisms, foods, are worse for you than non-GMOs. Okay? And very few people, about around 10%, believe they're better for you, even though a lot of the crops, a lot of the genes that are gene edited now are for that purpose, to increase nutrition, to make things more nutritious for us, or add a element of nutrition that is not available in that species before. Lots of those are around. The other components are, disease resistance, shelf life components, those kind of things. Okay? So let's look at one way that we can do that. CRISPR is a way that we can alter. Again, this is gene editing, going in, removing genetic material and replacing it with the gene that we're interested in. Okay? So CRISPR stands for cluster regularly interspace short palindromic repeats. Okay, big mouthful. That's why we call it CRISPR. It was discovered in bacteria. It's a way that bacteria can remove viral DNA, viruses that use bacteria to reproduce. Well, viruses will put their DNA into bacteria, and then it gets incorporated into the genome of the bacteria. Well, CRISPR is like a little pair of scissors that goes through and it can cut out those pieces of viral DNA, so they're not affected by the viruses. So when it was discovered, we then said, well, if bacteria can do it, can we use it to then tell it what place on a genome we need to cut, like this gene out, and then can we tell it what to replace it with? And indeed, modern technology shows that that's exactly what we can do. And it's getting more and more powerful to the point where we're potentially removing entire chromosomes. So if an individual has something like Down syndrome, and so they have 3 copies of chromosome number 21, we can remove that extra copy. And so it's very, very powerful tools that are going on. And so some of the things that we've been able to do is change things that, a lot of the, a lot of the fruits and vegetables that you might have in certain regions that you never see in stores is because their shelf life is so little. Like, You'd pick it, and 5-10 days later, it's no good. And that's not going to last for a store. A store is never going to want to have that unless we have, again, self-sufficient areas where everybody's buying local. Well, in some regions, it's just not possible. So one way that CRISPR has been used is to take some, fruits, vegetables, things like that normally we don't see, and get them into the market by altering their genes and making them less likely to rot, changing their fruit size, changing the number of fruits on the plant, okay, changing how the plant grows so it's more commercially friendly, lots of different things. Taking wild tomatoes, that might have different types of nutrients that when we bred them the traditional way, mixing this one and this one, that we got rid of a lot of the nutrients that are found in a lot of the wild type organisms. Well, get those organisms back into the markets by increasing their shelf life, or increasing how much fruit they might produce. Maybe they only produced a few fruit, so it wasn't worth commercial. Lots of different ways at which we have been doing it. Things that are notorious for having short shelf lives, or producing, browning that just is not going to allow people, or people don't want to buy because of that choice thing. We talked about choice before. Well, we can just delete out oxidase, which causes browning, and say like white button mushrooms and that kind of thing. This one's already on the market. Ground cherries already on the market. Rice, you guys have probably heard of like golden rice and others. So rice is very, very important, and so in some cases we're trying to increase grain yield, in other cases we're trying to add nutrients to rice that rice doesn't have, because it's such a staple in the world's diet. Okay, citruses, not allow for viruses to attack them, not allow for bacteria, all kinds of ways at which we can do this, which decreases the amount of pesticides, decreases the amount of chemicals we use on food fields, lots of things. Other things, knocking genes out, that makes it so things are resistant to tropical diseases. There's lots of things. Removing gluten. Okay, so if you need a gluten-free diet, generating wheat that has low gluten, or as little gluten as possible, so individuals don't have to alter their diet by consuming other species. And there's lists and lists of ways at which CRISPR has been used, and we're talking CRISPR's been around maybe for 2014, 2012 I think is when it was discovered. 2014 when it comes, so maybe 10 years, and now we have lots of things that are in stores, or proof of concept is happening with a lot of different species. So is technology the answer? I don't know. The technology was the answer that got us to 8.2, most people would argue. This is just one component of the technology. The other component is technology around medicine, medical field, technology around cleaning water, things like that. That transitioned us from 1 billion to 8.2. Is technology going to increase our carrying capacity so we can get to 12? We can get to 15? I don't know. But nonetheless, there are reasons to be hopeful with that, unless we as a population of people on the planet decide, okay, enough with exponential growth. We need to control our growth in a logical way and stop growing exponentially. I don't see that happening, but I guess it's possible. Otherwise, we'll be overshooting the carrying capacity, and then there will be a crash. And what that crash looks like, no one knows, but every population can reach carrying capacity. There just happens to be one population that may be smart enough to know what carrying capacity is and not overshoot it, and sit at carrying capacity for long periods of time. All right, so with that, we'll talk later.